All right, good morning, everyone. Welcome to Cycle 11. This is your last cycle for this course. This is the last week of lectures for biology. Please take the remainder of the time left to just catch up on whatever you need to in the course and focus on studying for the final exams so that you can hit a home run. Before we start lecture, I just have an announcement to make. There's a bonus grade that can be added to your overall course grade at the end of the course, 0.5%, if you complete a skill development survey. That is already available to you on the assessments tab. Just click on survey. It's not a very long survey. If you complete that, I will calculate your final grade and then just add 0.5%. Please do not underestimate this 0.5% because that can take you and bump you to the next level. We see that all the time. So that 0.5 can make a significant difference if you need to bump up to the next grade. This is the first time ever historically in first year bio that we're actually doing a skill development feedback. We've never had a skill development feedback. As course coordinator, I feel that I need more visibility in what's happening in labs. I don't have visibility in what's happening in labs. This is anonymous, so whatever you say is completely anonymous, so please be as honest as you can. The whole purpose really is to understand what aspects of lab component are working, what is not working, and how can we improve it. And students as partners is critical. I know a lot of faculty on campus that respect the students' opinion and their feedback, and I really think that sometimes when big decisions have to be made, believe it or not, I think having a survey of over 2,000 students and their opinion, I think some of those decisions have to be student-driven. So you guys have to be the drivers, not the passengers. And I really want that. I need your feedback. I need your help. I need your help. I need your feedback. And please answer it honestly. I need complete transparency to say, this did not work, this worked, but you need to change this. This is to help your future peers. So you can say, by the way, how's lab going for you guys? We did this survey and hopefully things have changed up for you guys. So hopefully they'll thank you for that. And I'm going to thank you for that. I have no visibility on what's happening. Okay. So what is today's lecture on? It is on epigenetics. What is epigenetics? When I taught this topic for the first time, it was actually in early 2000s. In a fourth year genetics class, of course, this is not the exact lecture, but the title of this lecture was The New Biology. And I was so proud to put that slide up. Guess what? The new biology. And everyone was, ooh, what's the new biology? And now here we are in 2026. This is the biology. It's no longer new. It's something that a lot of scientists had a lot of trouble getting on board with. And you'll see why. A lot of skepticism and, is this real? What is happening? Like, how can this be possible? Like real scientists, they needed evidence after evidence after evidence to be convinced. It's like, okay, finally, yeah, you convinced me. And then the articles just, they just exploded with all these articles on epigenetics and the effect that it has on our gene expression. So what is epigenetics? Epi means on top, and genetics is your genomic sequence, those bases, those nucleotides, A, T, C, G, G, C, A, T, all those bases that make your DNA sequence, that is the genetics part, the genome. So you have a genome, right? You know that. You've studied that for so many months. Epi, meaning on top, is referring to all the tags that are on top of your genetic sequence. So epigenetics. You have a genetic sequence and you have an epigenetic sequence. So you have a genome and an epigenome. They're actually different. The epigenome are the tags on top. So what are those tags? Those tags are methyl groups and acetyl groups. Methyl looks like this, a CH3, and methyl groups are added to cytosine nucleotide, not to other nucleotides for purposes of this course, but this is the foundation. Like you really don't have to know a whole lot more to build on this. So methyl groups are added to the cytosine nucleotide on the DNA itself. Acetyl groups are added to histones. What are histones? Histones are proteins. They're like a complex proteins. Several small proteins make up one large complex. It's called the histone. The histone helps to wrap DNA. It spools the DNA so that it's compactly stored in your nucleus. DNA has an overall negative charge. Because of the phosphate backbone, it has an overall negative charge. So if you have to take that long piece of DNA, if you stretch out your DNA, it's very long. You need to compact that. So think of a spool and a thread or a yarn and just spooling it, but you have a bunch of spools. They all come together and they spool all that thread. So that's exactly what's happening here. So if DNA has an overall negative charge, then what overall charge do you think histones would have? Positive. Positive, negative, attract. So because histones have an overall positive charge, or at least a good component of it, it's able to grab that negatively charged DNA and spool it and compact it. So you see this histone here? That's several proteins. Histones have histone tails. Tails are just polypeptides, like amino acids that have not folded into the protein structure yet. It's just, that's how the structure is. It just sticks out. So it's still part of the protein. It's just not taking a structure. It's just taking it's still a polypeptide chain. So it just sticks out of it. It's a tail. It's part of the histone. So methylation is when cytosine gets methylated. That's your tags, one of your tags. It's called an epigenetic tag. The other tag is acetylase. So acetylase gets added to the amino acid called lysine. Lysine has a positive charge. So amino acids are categorized into different types: positively charged, negatively charged, hydrophobic, hydrophilic. And so lysine falls in the category of positively charged amino acids. And that's really where histone gets its positive charge, is through lysine. So lysine is present on its tail, including inside the protein as well, but the one that's pertinent to epigenetics is the one that are exposed on the tail, lysine, positive. When you add an acetyl group to it, or you acetylate it, that's the acetyl group that I'm circling up on top there. When you add that, lysine loses its positive charge and it becomes neutral. So you're going from this positive to a neutral state. So when you add a methyl group, that's called methylation. When you add an acetyl group, that's called acetylation. Did you guys know that this methylation and acetylation is dynamic? It's reversible. It can go the other way also, and it can change throughout time. So when you remove the methyl group, it's called demethylation. When you remove the acetyl group, it's called deacetylation. So whenever you remove, just add the letters DE, that's D. So methylation, demethylation, acetylation, deacetylation. So these tags go on and off. What are these tags doing? They are controlling gene expression. So in case now you're like, wait a second, what happened to cycle 7 and 8 that I so painfully had to sit through, where you talked about promoters and transcription factors and promoter proximal and enhancer regions and negative regulators and positive regulators. Oh my goodness. So they don't regulate gene expression? They do. But the epigenome decides when, where, and how much gene regulation is going to take place. So what determines whether these tags go on or off, when and where, and how much? Do you know what decides that? Your personal life experiences. As you go through life, these tags are changing in your genome. Your epigenome is changing as you live life. So any decisions you make, anything that you encounter in life, any stimulus, environmental stimulus, it can be in any form, any shape or form, that affects your epigenome. Your genetic sequence, not so much. You're going to have sporadic mutations as you go through life, of course, but your genetic changes are not that drastic. If you look at your your genomic change from birth till now, you guys are quite young, it won't be that drastic. You're going to have sporadic mutations because you're still living life, right? Depending on your lifestyle. But your epigenome, it's already different. It's already, I would say, significantly different. Not drastic, but it would be very significant. This is the relationship. Now, I never like to use a gun as an example in my lectures, but we're all adults and you're going to read about it on the internet anyway, so I might as well tell you. Scientists use the gun as an example to understand the relationship between the genome and the epigenome. If you look at the gun, the bullets are your genetic sequence. Those are the bullets, your genome. But it's the epigenome that pulls the trigger. So understand that you can't have one without the other, but it's really the epigenome that's determining when, where, and how much gene expression are you going to get. And once that's determined, then all the factors that you learned about come into play. The gene is completely exposed, and now everything that you learn in cycle 7 and 8 takes place. So one of the best studies, and many, many studies have been performed on this, some of the best studies that really have sealed the deal for the role of the epigenome and how it changes and how it's dynamic and the effects it has on gene expression have been from studies done on monozygotic twins. So monozygotic twins are born with identical genomes. But if you look at their epigenome, just starting at the age of 2 years, you can see a significant difference. But guess what? Studies have shown that you can see a difference in utero. Depending on the position of the embryo in the uterus, how much food is it getting, like how much oxygen, like little things like that actually start to show subtle changes in the epigenome. So what does that tell you? That tells you that the epigenome is sensitive. It's sensitive to change. It's sensitive to what you bring into yourself from the environment. I saw an interview once of 70-year-old twins. They had their epigenome sequence. So you can get your methylome. Methylome is like, how you have a genomic sequence, you can have your methylome, or your epigenetic sequence. And methylation tells you more than acetylation, which is why they do the methylome. And a lot of people have had their methylome sequence, a lot. It tells you far more than your genome, to be honest, far more. So they got their methylome done. And they noticed that there was a drastic difference in their epigenetic sequence, but not so much in their genetic sequence. And so when they're interviewing them, they were quite shocked. They were really surprised. And they're not in science and they didn't quite understand what's epigenome, what's genome. But all they knew, and they felt that, oh my gosh, like this is shocking. I don't understand. How come our DNA looks different? We are identical twins. And so in the middle of the interview, one of the twin sisters, she started to cry. This poor 7-year-old woman, she started to cry. And the interviewer asked her, said, why are you crying? And she said, my whole life, I thought that my twin sister was genetically identical to me. Like that's the one thing we had in common. And that's the bond we had. We led separate lives, different parts of the world. But that's what kept us together, was that we were genetically identical. And now you're telling me that we're not genetically identical. And the interviewer said, you're still genetically identical. You're just epigenetically not identical. And she said, epigenetically not identical? We're epigenetic opposites. And they were. They were very, very opposites. They're like areas that were hypermethylated or hypomethylated, it was the opposite in the other twin. So it's just, you go through life, you make different decisions, right? Little decisions make a difference. The studies that have really marked this type of evidence and shown that experiences can really have an effect are really looking at children, in their formative years 1 through 5. That's been really, really eye-opening, that how strong the epigenomic marking is, because you really see the world in a different way, and I think you perceive things in a different way between 1 to 5. So that marking is very different. It's not as reversible as other markings later in your life. So it becomes less reversible. And that may be a good thing or a bad thing, right? It just depends. Other studies that have brought a lot of light are people that have similar trauma in life, they have a similar epigenetic pattern. So the studies that showed a lot of evidence were children that underwent child abuse. Child abuse victims actually share similar epigenetic patterns. And this is looking at victims across the world, from Africa to Asia, Europe, North America. So what are the chances? I mean, they're living in different environments, different families, but they have similar epigenetic markers because they share that same traumatic experience, which is being abused as children. Let's talk about something happy and positive. If people that are happier and smile more and just see the glasses half full are more optimistic, I'm not kidding, guys. You can Google this stuff, right? Does it sound like science to you right now? Are you guys sitting there going, did she go off the rails? Like, thank God this is her last lecture. But it's, no, this is real. This is why it took so long for people to figure this out. This doesn't make sense. As a scientist, you want proof. This sounds really hokey. This sounds it sounds silly. It doesn't make sense. No, it is true. And you guys can Google all the papers in the last 20 years. So people that smile more and see the glasses half full, and they have a similar epigenetic pattern. And that pattern seems to be associated with the neurotransmitters. They connect with that. Serotonin and dopamine, you guys can Google what these things do in your brain and how you feel and how you react. They actually upregulate and downregulate genes that react with these neurotransmitters as well. So there is a scientific connection. So this is not just hokey, people coming up with these random theories, scientific evidence. And people that are not happy and seeing the glasses half empty all the time and pessimistic, they also share an epigenetic pattern. So this is not coincidence, right? It's not just coincidence. So there is something there. It's how you think, how you feel, your thoughts, your actions, your words, everything makes a difference. So yeah, guys, just, make good decisions. The last thing I want to see in this slide is that your epigenome is heritable. You can pass, or you pass your epigenome to your offspring, and you have inherited epigenome from your parents. Your germ cells have an epigenetic sequence. Why would they go away? They stay in the haploid cell. When it gets fertilized, though, that's another story, because the epigenome needs to be there for development because they need to control gene expression. But I'm not going to go into too much detail what happens during embryogenesis. So in case there are some things that are missing for you, you can come and ask me. I can explain who, what, where, when, how do you inherit it? So understand that your epigenetic sequence right now comes from two sources. Number one, from your parents. So from one parent and the other parent, they both have contributed to that zygote. And the second way that it comes is through your personal life experiences from birth up until now. So whatever decisions you've made, you've marked your genome that way, and now you'll continue to mark it, and then whenever you have children, at whatever point you have them, you will pass them on at that stage. So things like habits and behavior and thought process, believe it or not, you can actually transfer that to your children, which there are even a lot of just syndromes and even behavior. A lot of psychologists are looking at epigenome because you're trying to understand there's no gene for a specific behavior, right? There's no gene for the way you act. There's no gene for the way you smile or something. It's your thought process. Sometimes do you guys find you think a lot like your parents? Like, I know I do. I have that. I have certain habits. And then others will argue that's because you learned that behavior. You lived with your parents for so many years. You learned it. It's a learned behavior. Maybe it is. Maybe some of it is. Maybe some of it isn't. This whole nature-nurture thing has changed. The debate is now different. It's different in the sense that when I took psychology many, many years ago, decades ago, first year psych, they talked about the nature-nurture. It was such a big topic. Oh, is it nature? Is it nurture? Is it nature? What do you guys think? How much is nature? How much is nurture? The bottom line is a lot of the nurture is now being equated to nature because the sequence is changing based on the stimulus. Like, they have shown these studies in animals over and over again and in humans over and over again, like for 2.5 decades, guys. Like, it's no longer the new biology. Oh my God. So it's been shown. So yeah, it's if you want proof that nurture has an effect on who you are and what you react to, and whether you get cardiovascular disease later in life or not, whether you get diabetes or not, all this is linked, guys. So whether you get inflammation or not, whether you have a lower immune system or not, whether you have anxiety or not, all of this stuff I'm saying is all linked. It's all studies. You can read them. They're all linked. So if you have any doubts, then the doubt can go away if you need proof. The proof is in the epigenome. The proof is there. So you can look at punctuation as an analogy to epigenetics. And these words are your genomic sequence. So all of those letters are the same. The difference is punctuation. So where you have a comma, where do you have a period, and where do you have capitalization. That changes the sentence completely to have a very completely opposite tone, opposite meaning. It's the exact same thing, guys. You can dictate how you want to live life and how you want your genes to be expressed, which ones, where, and how. You actually have more power than you think. Back in the day, it was, my gosh, one gene, one protein. We went from that, one gene makes one protein, from that to look how far we've come. Look how far we've come to say that, everyone has those genes. Everyone makes the best out of those genes, and you decide how you want to do that. It's really about making good decisions for the most part. And some articles argue it's like, well, you're stuck in that environment. What we go and breathe is we can't help it. You go to the grocery store, how many things can you buy that are really truly just, you can say, yes, there are no preservatives in here. There's nothing bad in here, right? This is, you pick up a broccoli, you got problems there too, right? So I'm just saying that a lot of things are not in your control, but things that are in your control, yeah, why not? Another way to look at epigenetics is if you have your textbook without any sticky tags, that's your genetic sequence. That's already there. It's done. Like, that's your book. Here you go. But if I gave this book to everybody in the classroom, but I put specific sticky notes for each individual and said, you only have to read the yellow sticky notes, you only have to read the pink sticky notes, you're only reading certain chapters, certain sections. So I can't give you all the same midterm exam, right? I have to give you separate midterm exams based on what you read, based on what the experience you have of reading. I can't test you on something that you haven't done. So you have to understand that you are building your epigenome to be able to write a certain midterm exam, to be able to write a certain final exam. So this slide may be very obvious, it's very intuitive. Yes, Dr. Sharan, I get it. All of these things influence my genetic sequence and my epigenome. The obvious ones, toxic chemicals, drug abuse, UV radiation, etc., your diet, of course, all of these things, your psychological state, they all have an effect on your genetic sequence. So yes, these ones far more, your diet as well. Exercise has a positive influence on your genetic sequence. So those are obvious. What may not be so obvious is that all of these things have a very significant influence on your epigenome, and it's quite sensitive. You don't have to be doing drastic things to see drastic changes in your epigenome. In your genetic sequence, yeah, you really have to be living life on the crazy side for your genome to be looking completely different on a regular basis, because I think you'll eventually die very soon. Your genetic sequence will protect you, but it's your epigenetics that actually controls gene expression. So control the epigenome, guys. Like, focus here. And so yeah, these are quite obvious, but you know what really spiked a lot of interest in epigenetics and like eyes open wide was social interactions. Scientists showed that who you socialize with, the people in your life, they can change your epigenome. And without you even realizing, you're not cognizant of it. You're not like, oh, you know what? This is happening. But you're feeling sometimes the effects. The effect could be positive. The effect could be negative. Does everyone have that person in their life that just sucks the energy out of you? Or just like, I don't know, you spend time with them and then you're like, I don't feel so great after the words, but you're like, oh, it's not them, it's me. Because we're all adults, right? You take ownership, you take accountability, you don't blame anyone for anything. If you don't like them, don't hang out with them, right? I know you can't choose your family, but still, it's like you manage, right? But just understand that it may take a while for you to realize that maybe this person had a positive influence or a negative influence. So just be mindful. That's all I'm saying. Just don't go and befriend everybody or not befriend everybody. Just be mindful of, is this having an effect on my psychological health? Yes or no? Just be mindful of it. So we are in a biology class, so we've understood what epigenetics is, how it influences gene expression, what do we have to do to possibly control our epigenome, and the effects that it can have on our psychological and physical health. So now what we're going to do is look at how these tags are put on and how are they removed. And if we understand this, you can now design drugs. Did you guys know that some of the new drugs that are coming out are epigenetic drugs? They're actually changing the epigenome instead of the genetic sequence. So you know how we were talking about CRISPR-Cas9 going and having an effect on the genetic sequence? Next class, you'll learn about dCas9, which is dead Cas9, meaning it doesn't have any catalytic activity. It doesn't cut the genome at all. So you're like, so what's Cas doing then? Cas is a nuclease, it cuts, right? But what are we using Cas for? To piggyback enzymes. It takes it with the guide RNA to the target, but guess what? It leaves the genome alone, and it targets the epigenome. It methylates, demethylates, acylates, deacylates. It's so awesome. It's cool. So this is really where this new CRISPR-Cas9 is taking us. So what are the enzymes? The enzyme that methylates the cytosine nucleotide is called DNA methyltransferase. You don't have to write this down. All the long form is written in all the subsequent slides. DNMTs, it's in the name. These are methyltransferases, so they transfer the methyl group to the DNA methyltransferases. TET is another protein that removes the methyl group. So DNMTs add, they methylate, and TET demethylates. You need to do both because our tags are dynamic. They're going on and off depending on your life experience. So maybe you make a decision, it goes on. You make another decision, it goes off. And it's right now, as we speak, it's probably happening in people's bodies right now, literally, as we speak, like methyl tags going on, methyl tags going off. Histone modification are done by HATs and HDACs. What are HATs? Histone acetyltransferases. These are enzymes that transfer acetyl group to histones. It's right in the name, in case you're ever confused. What do HATs do? Just remember what it stands for. HDACs are histone deacetylases. So these are enzymes that remove the acetyl group or deacetylate histones. So DNMTs, TET, HAT, and HDACs. Here you have your genetic sequence, also known as the hardware. When that sequence changes, that is a mutation. We consider that to be a mutation. And as you know, a mutation can be a good thing or a bad thing. We don't know, it just depends, or it could be nothing. So this is a mutated DNA. This case, you get epigenetic chain, which is also known as a software. If you want to look at some analogies here as a software, your DNA is unmethylated, but then it becomes methylated. So a DNMT comes and it methylates it. This is not a mutation. This is just an epigenetic change. We don't refer to that as a mutation, a methylation mutation. There's no such thing. It's just an epigenetic change. So the definition of epigenetics, the study of changes in gene expression caused by mechanisms other than changes in the underlying DNA sequences. Genes can be tagged by our experiences. What are those tags? Methyl groups and acetyl groups. This is a dynamic process, as we discussed. It keeps changing through your life. This is not random. So if it was random, then people that were happy or sad or abused or had any traumatic life experience would not have the same tagging. Tagging is not random. So it's quite programmed. So if you have this traumatic event, they've shown that these are the tags that come with it. These are the genes that get expressed with it. These tags can be inherited by the offspring, and that is known as transgenerational epigenetic inheritance. So you do pass on your tagging to your offspring. So if that's one reason for you guys to be more mindful of what you do, just understand that some of the tagging that's happening, it'll be passed on to your next generation. So now let's turn to more details, because we're in biology and we like molecular biology, right? You have your cytosine. DNMT comes in, DNA methyltransferase. It methylates the cytosine. That's DNA methylation. How and where does it happen? Well, this is how it happens. You have this enzyme. It's an enzymatic reaction. It simply adds a methyl group to this carbon here, carbon 5. But what are the regions of the DNA that are actually methylated? They're called CpG regions. CpG. CpG. P means the phosphate group between them. Because C and G also complementary base pair, right? So if they're across from each other, there's no P between them, but if they're next to each other, there's a P between them because they're part of the backbone. The P is part of the backbone. So CG. If that's CG, then the next strand, 3 to 5, is going to be GC due to complementary base pairing. So when this cytosine is methylated, this cytosine is also programmed to be methylated. Methylation always looks like this. It's not just the one strand. DNA is a double strand. That's the structure of DNA. So if one strand is methylated, so is the other. And those are in CpG regions. When you have a bunch of CpGs together, that is known as a CpG island. They call it an island, with the analogy that your entire genome is the ocean, and then you have islands, and those islands are CpG regions in your genome. So what do you think these islands are? Where do you think they are? If you had to guess where these islands were, what would you guess? The epigenome is controlling gene expression, so it's not going to do something totally crazy and wild. Yes? Close to a promoter is a very good answer. They are on the promoter. These CpG islands are actually the promoters. The promoter regions themselves, CpG. So when, see these black symbols here, these are methyl groups. The white or clear are non-methyl groups. So there's no methylation on that cytosine nucleotide. So when you get hypermethylation, so you get lots of methylation, the gene transcription is off. When you get hypomethylation or unmethylated CpG regions on the promoter, the gene transcription is on. So methylation inactivates transcription, and demethylation activates transcription. If you look down here to the bottom left, you can see these pink are methyl groups. On the promoter, transcription factors cannot bind. So methylation of cytosine nucleotides prevents transcription factors from binding, therefore transcription cannot occur. This is how methylation inactivates transcription. We'll get to the bottom right here in a bit, but let's go to the top here. How do you get acetylation and deacetylation, and what effect does that have? Well, when the histone tail is not acetylated, that means that there's no acetyl group on it. That means lysine is positively charged. So histone has a really tight grip on the DNA. And that state is known as the heterochromatin state, tight, condensed and tight. The gene is not accessible. The pink here is the gene that needs to be transcribed and translated. It's not accessible, so therefore the gene is inactive. So it's the acetylation of the tail and deacetylation that dictates whether this histone is going to be tightly grabbing the DNA or not. So in this case, it's tight because the lysine is positive. Positive and negative like each other. In this case, however, when you get acetylation, meaning an acetyl group is added, then what happens? The lysine is now neutral. When it's neutral, it lets go of the DNA a bit. That positive-negative attraction is not so strong. It loosens up. That loose state is called euchromatin. So in a euchromatin state, look what happens. the gene is exposed. That DNA portion that codes for all the proteins and RNA that you need is exposed, it is accessible, and now everything that you learn in cycle 7 and 8 happens. Promoters, transcription factors, like splicing, alternative splicing, everything that they need to do to make that protein or that RNA is happening. But who allowed it to happen? Who said it could happen? The epigenome. So now look down here. You have your methylated promoter. To the methylated promoter, what binds is methyl binding domain proteins. Proteins that can have their domain or a region that recognize the cytosine methylated form. When that happens, then that protein, MBD, it recruits HDACs, histone deacetylases. It recruits it and says, come, condense this even more. Like, look at that. No transcription factor binding. And now this in itself recruits the HDAC. And now HDAC says deacetylate. Deacetylate means to remove the acetyl group. If you remove it, you have the heterochromatin state. So now in this heterochromatin state, you don't get transcription. So methylation and acetylation, they're working together. Now let's go into detail of methylation and demethylation. We're going into more and more detail each time. A little bit of added information. It's not drastic. Here you have your double-strand DNA, and this has to be methylated. Why? Well, de novo means new. Nothing is methylated here. Brand new methylation, guys. Let's put it on. An example that we learned, you have a life experience. Now you're going to methylate your promoter, whereas someone else does not have that experience, is not going to methylate their promoter. It could be twins. One twin is doing it, the other twin is not doing it, depending on what that twin is doing. So this is de novo, from new. DNMT3A and 3B, they're involved in de novo, so new. Now, does your DNA replicate? Do many cells in your body replicate? Oh, yes. So are you going to lose methylation as your DNA replicates? No, you're going to maintain the DNA methylation. There is a way to lose it through synthesis, but we're not covering that. Just understand for this course that methylation is maintained. It is maintained. It has to. You have to maintain the methylation in the daughter cell. If methylation is regulating gene expression, you can't say, well, once my cell divided, I lost methylation, and so I don't know what's happening to my genes now. No. It doesn't work like that. Your daughter cells are identical. So they're going to also have methylation. But this is what happens. You get DNA synthesis. Both of these methylated strands, they serve as template. Then your new strand does not have methyl groups because DNA polymerase doesn't grab a cytosine that's methylated and then adds it to the growing strand. That's not how it works. Cytosine is not methylated. It grabs the cytosine, it puts it on, and then the methylation happens after synthesis is complete. So it's a separate process. So now DNMT1 is the enzyme that recognizes hemimethylated strands. Hemi means have. That's what is programmed to do through evolutionary time. It goes, here's one that's methylated, here's one that's not methylated. I know what I have to do. I have to copy the methylation over. It just copies. It goes CPG, 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 got it. It sees a C, G, boom. This is methylated, crisscross, methylate, crisscross, methylate, crisscross, methylate. It just has that memorized. It just boom, 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 it does it. So now you have maintained your methylation. TET removes methyl groups. When would you have TET removing methyl groups? Life experiences again, right? As you go through life, you're going to remove those tags as well. Put them on, remove them. So some of you will be removing it for certain promoters. Some of you will be putting on it for certain promoters, depending on what your behavior is, what your stimulus is, how are you seeing the world. So that's going to be TET coming in to remove. And these enzymes end in ASE, except for TET. TET doesn't really tell you what this enzyme does because they still kept the same name based on how it was discovered. It was discovered during a chromosome 10, chromosome 11 translocation during cancer, so they kept the same name. So don't get confused. What is that translocation 10, 11? It's just a name. Just know it as TET I won't ask you to ever write the full form. So now let's go into a little bit more detail of histone acetylation and deacetylation. Here you have your histone molecule. That's what it looks like in blue and green. And the DNA is wrapped around it. This is just one spooling. You have a whole bunch of these happening in your DNA. So the blue is actually an octamer. It has H2A, B, 3, and 4, each one twice. So 4 at the top, 4 at the bottom, that octamer, plus this histone 1, that is actually your histone molecule. And these tails sticking out of it, that is part of this blue that I'm circling there, right? That's part of it, except that that one is not folded into that protein structure. That's just sticking out as a polypeptide chain. It's called the tail. So now you have this DNA that wraps around it. The core DNA comes around it, and then you have this DNA sticking out. That's linker DNA. What does it do? It links the next, the next, the next. So that's how it's all spooled all throughout your genome. This is just a blow up. So this is your histone with your core histone with your tail sticking out. The tail is now this right here. These are the specific amino acids. So these letters are all different amino acids. All amino acids have a letter denoting them. K stands for lysine. So K in the position 5, position 8, 12, and 16 here are the ones that are going to get acetylated or deacetylated. Now, do all of them get acetylated? No. Do some of them do? Yes. Could it be all? Yes. Depending on the cell type, depending on your experience, it depends on a bunch of things. So when you read these articles, oh my God, these poor scientists have to come up with like, which site does what? It's all through empirical data. It's a lot of work, guys. The bottom line is these all have the ability to be acetylated and deacetylated. How? You have your lysine, positive. See that positive? That's the natural structure of lysine. That's how it exists at our pH. pH 7, that is the lysine, a positive charge. You get the addition of the acetyl group at the end here. That is acetylation. Acetylation. When you get acetyl group added, it's acetylysine. Lysine now is neutral. So what do you get? You get euchromatin, right? Loose. So you get activation of transcription. Deacetylation, what does that mean? You remove the acetyl group, go back to positive, it has a tight grab on the DNA, heterochromatin state, no transcription. So the way some students memorize it is acetylation is activation of transcription, and deacetylation is decrease in transcription because it has the word D, DE. If you want to memorize it that way, that's fine too, if it's going to help you on the final exam. So here you have the slide, again, showing you the exact same thing. I just want to point this out again. Lysine, positive charge. Acetylation by HATs. HATs are the ones that are acetylating. Histone acetyl transferases. They're transferring acetyl to histones. It's in the name. So when they do that, they're acetylating. What does that lead to? Increase in transcription. Deacetylases, they're removing acetyl groups from the lysine or the histone. So deacetylases, that decreases transcription because if you remove it, guys, lysine is back to positive, has a tighter grip on DNA, heterochromatin state, the gene is not accessible, decrease in transcription. Okay, so just nothing to memorize, just understand it. So here's a slide, a summary of what we discussed today. Histones get modified and your DNA gets methylated. Histones gets modified by acetylation and deacetylation. DNA gets methylated by methylation and demethylation. And the enzymes involved are HATs, HDACs, DNMTs, and TET. So we have 5 more minutes. So what I'm going to do is spend that time, and I'm going to give you some ideas on how I'm going to ask questions on this specific topic, not epigenetics, but this slide here on the exam. You are responsible for everything that we discussed in slide 1. I was going on and on. You're responsible for all of that stuff. So don't be surprised if that stuff comes on. I want you to understand the consequence of overexpression of any of these enzymes. So if HATs are overexpressed, if HDACs are overexpressed, or underexpressed, what will be the outcome? You just have to work it out. And I would want you to know the outcome in several ways. In the euchromatin or heterochromatin state, I would want to know if lysine is positively charged at that point or neutralized. And I would want to know if there's an increase or decrease in transcription. It could be a multiple. So all of them could be correct. I'm already telling you how I'm going to ask the question, guys, so I'm making your studying a lot easier. So if you have overexpression, we're taking one step further, guys. How are these enzymes made in the cell? Through transcription and translation. These are proteins. These are all proteins. So it involves transcription and translation, and each one of these has their own transcription factor to make them. So if I overexpress the transcription factor for HATs, what's the outcome in the cell? If I overexpress transcription factors for HATs, that means you have overexpression of HATs. You have more HAT in there. If you have a lot of histone acetyltransferases, then what's going to happen? Are you going to get an increase in gene expression or a decrease? Overexpression of HATs is going to lead to increase. Good, I'm seeing a lot of students go increase. Beautiful. An increase in gene expression. Now, you might say, well, that's because I memorized HAT. HAT increases expression. Sure. You might get one of the multiples and go, oh, well, 2 is correct. Then obviously, you'll be able to work it out. Sure, maybe, but don't do that. Because the next option could be, so what is the state of lysine if I overexpress HAT? Most of the lysine is neutral. Because HAT adds the acetyl group. It's now neutral. And what is the state of the DNA at that point with HATs? With most of the regions would be in, if you're overexpressing it in the cell, you have lots of HATs. Like the cell's not really normal to have that much histone acetyltransferases. What would be the state of the DNA? When I say state, I'm talking about euchromatin or heterochromatin. Lots of HATs lead to euchromatin. Very good, euchromatin. Euchromatin, open, exposed. Now you get transcription. So this is the way I'm going to ask the question. So I think that should be pretty helpful. All right, yeah, it's the end of today's lecture. See you guys next class. Thank you.